Good morning, everyone. The topic of my presentation is neuropeptides in psychiatry, and my moderator is Dr. Sanjeev. Here we'll be studying about the introduction of neuropeptides, their biosynthesis, distribution and regulation, signaling, thyrotropin-releasing hormone, corticotropin-releasing factor, oxytocin and vasopressin, neurotensin, and the role of neuropeptides in various psychiatric illnesses. First of all, the introduction. Neuropeptides are the most diverse class of signaling molecules in the central nervous system. Initially discovered for their role in hypothalamic regulation of pituitary hormone secretion, peptides have been found to play a complex role in brain function over the last 35 years. Neuropeptides and their receptors are widely distributed throughout the CNS, where they exert an extraordinary array of direct or neuromodulatory effects, ranging from modulating neurotransmitter release and neuronal firing patterns to the regulation of emotionality and complex behaviors. Here is a list of various neuropeptide transmitters that are ACTH, angiotensin, apelin, atrial natriuretic peptide, bombesin, calcitonin gene-related peptide, cocaine and amphetamine-regulated transcript, cholecystokinin, corticotropin-releasing factor, dynorphin, beta-endorphin, du-enkephalin, met-enkephalin, galanin, gastrin, ghrelin, gonadotropin-releasing hormone, growth hormone-releasing hormone, insulin, kisspeptin, motilin, neuropeptide S, neuropeptide Y, neurotensin, orexin, oxytocin, prolactin, secretin, somatostatin, urocortin, thyrotropin-releasing hormone, vasoactive intestinal polypeptide, and vasopressin. Now, what is a neuropeptide? A neuropeptide, it is a chain of 2 or more amino acids linked by peptide bonds and differs from other proteins only in length of the amino acid chain. Neuropeptides range in length from 2, example carnosine and N-serine, to over 40 amino acids, example corticotropin-releasing factor and urocortin. By convention, peptides greater than 90 amino acids in length, about 10,000 molecular weight, are considered proteins. Here, it is the amino acid sequence of various neuropeptides. First one is thyrotropin-releasing hormone, then corticotropin-releasing hormone, arginine, vasopressin, oxytocin, neurotensin, and neuropeptide Y. Neuropeptides have been implicated in regulation of a variety of behavioral and physiological processes that include thermoregulation, food and water consumption, sex, sleep, locomotion, learning and memory, response to stress and pain, and emotion and social cognition, etc. Involvement in such behavioral processes suggests that it is likely that neuropeptidergic systems contribute to the symptoms and behaviors exhibited in the major psychiatric disorders, including autism, schizophrenia, mood disorders, and anxiety. Now, the biosynthesis of neuropeptides. This involves transcription of an mRNA from a specific gene, translation of a polypeptide pre-hormone encoded by that mRNA, and then post-translational processing involving proteolytic cleavage of the pre-hormone to yield the active neuropeptide. Neuropeptide genes are generally composed of multiple exons that encode a protein pre-hormone. The N-terminus of the pre-pro hormone contains a signal peptide sequence which guides the growing polypeptide to the rough endoplasmic reticulum membrane. The single pre-pro-hormone molecule often contains the sequences of multiple peptides that are subsequently separated by proteolytic cleavage by specific enzymes. Here are the gene structure, the pre-pro-hormone mRNA and processed neuropeptides of thyrotropin-releasing hormone, corticotropin-releasing hormone, oxytocin, vasopressin, and neuropeptin. Here the boxed regions, these are the locations of the exons in the respective genes, and the shaded region, these are the coding regions. Each pre-pro hormone begins with a signal peptide sequence SP. The black boxes here, these indicate the location of the sequences encoding the neuropeptide. The mRNA encoding the pre-pro hormone is translated by ribosomes associated with the rough endoplasmic reticulum, and the growing polypeptide is translated into the cisternae of the rough endoplasmic reticulum with the signal peptide anchored in the rough endoplasmic reticulum membrane. Once translated, the signal peptide of the pre-pro hormone is cleaved by a single endopeptidase, freeing the pro-hormone polypeptide. The pro-hormone is then shuttled to the Golgi apparatus, where packaging into granules or vesicles occur. Proteolytic cleavage of the pro-hormone into the biologically active neuropeptide begins in the Golgi and continues in the granules. Production of biologically active neuropeptides from pro-hormones begins with cleavage at specific sites adjacent to the neuropeptide sequence by specific endopeptidases, which are known as pro-hormone convertase. Pro-hormone convertases cleave generally at pairs of basic amino acids, example lysine-arginine or lysine and lysine, and arginine-arginine, flanking the neuropeptide sequence. Pro-hormone convertases are co-packaged with the pro-hormone in the granules of the Golgi apparatus. Following endopeptidase cleavage, the peptide fragments are subjected to exoproteolysis by carboxypeptidases or aminopeptidases in order to remove the residual basic residue or the carboxy or amino terminus of the peptide fragments. Here we can see the biosynthesis of the peptides. This is the exon terminal, and here calcium channels are, calcium is present. Here in the Golgi apparatus, the main conversion from the pre-pro hormones to the pro hormones occurs, and the granules are stored in the here. This is the granule pool. Then through the exon, they travel to this storage pool towards the exon terminal, where the calcium ions are present. Now the distribution and regulation of peptide. The peptides involved in regulating pituitary secretion are concentrated in the hypothalamus. Hypothalamic releasing and release inhibiting factors are produced in neurosecretory neurons adjacent to the third ventricle that send projections to the median eminence, where they release peptides into the hypothalamo-hypophyseal portal circulatory system. Peptides produced in these neurons are often subject to feedback regulation by the peripheral hormones that they regulate. For example, the thyrotropin-releasing hormone regulates activity of the hypothalamic pituitary thyroid axis, and thyroid hormones negatively feedback on thyrotropin-releasing hormone gene expression. However, neuropeptide expressing neurons and their projections are found in many other brain regions, including the limbic structures, cerebral cortex, midbrain, hindbrain, and spinal cord. Neuropeptides are often co-localized and released with other neuropeptide or non-peptide neurotransmitters, refuting the one neuron, one transmitter tenet. The co-localization of neuropeptides within classical neurotransmitter circuits suggests an interaction between these systems, and the modulation of monoamine neurotransmitter, example norepinephrine, function by neuropeptide, is common and well established. Indeed, many neuropeptides are co-transmitted with non-peptide neurotransmitters and serve to modulate neurotransmission of both the pre- and postsynaptic cells. The signaling of neuropeptides. Neuropeptides act as neurotransmitters, neuromodulators, and neurohormones. Their release is not restricted to synapses or exon terminals and occur throughout the axon or even from dendrites. In the end, they are released by exocytosis of the granules in response to electrical or hormonal stimulation of the neurons. Stimulation of these neurons increases intracellular calcium concentration, which leads to the fusion of peptidergic granules to the plasma membrane, releasing to the expulsion of the peptides into the endothelial cells. Specific neuropeptide receptors conciliate the cellular signaling of the neuropeptides. Most of the neuropeptide receptors are G protein coupled 7 transmembrane domain receptors belonging to the same family of proteins as monoamine receptors. Each neuropeptide receptor is specifically coupled to one type of G protein, either GS, GI, or GQ, depending on the subtype of G protein with which the receptor interacts, which may result in stimulation or inhibition of specific second messenger pathway. The most common signaling pathway involving activated G protein modulating the activity of either adenylate cyclase or phospholipase C. The first neuropeptide is thyrotropin releasing hormone. In 1969, thyrotropin releasing hormone, a pyroglutamyl histidyl prolinamide tripeptide, became the first of the hypothalamic releasing hormones to be isolated and characterized, work that in part resulted in the Nobel Prize of Dr. Andrew Shelley and Roger Gilman in 1977. Thyrotropin releasing hormone is widely distributed in the CNS, with thyrotropin releasing hormone immunoreactive neurons being located in the olfactory bulbs, entorhinal cortices, hippocampus, extended amygdala, hypothalamus, and midbrain structures. The discovery of the structure of this hormone led to the conclusive demonstration that peptide hormones secreted from the hypothalamus regulate secretion of hormones from the anterior pituitary. The gene for TRH in humans resides on chromosome 3q13.3-q21. Corticotropin releasing factor. In the 1950s, it was observed that pituitary extracts contained a factor referred to as corticotropin releasing factor, which could stimulate the release of ACTH from anterior pituitary cells in vivo. After a search spanning nearly 3 decades, Weil and colleagues isolated the characterized corticotropin releasing factor as a 41 amino acid peptide in 1981. The gene for it is present in chromosome number 8q13 in humans. Most recently, the related neuropeptides, urocortin 1, urocortin 2, and 3 have been identified and share similar gene structures. There is convincing evidence to support the hypothesis that corticotropin releasing factor and the urocortins play a complex role in integrating the endocrine, autonomic, immunological, and behavioral responses of an organism to stress. Now, here is the corticotropin releasing factor system. It is further complicated by a fact that effects of corticotropin releasing factor and the urocortins are mediated by at least 2 of the receptor subtypes, that are corticotropin releasing factor 1 receptor and corticotropin releasing factor 2 receptors. This is the darker the arrow, it shows it affects more of that receptor, and lighter the arrow, it shows its less effect on that receptors. So we can see a corticotropin releasing factor, it has more effect on 1 receptor and less effect on type 2 receptor. Urocortin 1, it has less effect on type 1 receptor and more effect on type 2 receptor. While urocortin 2 and 3, they act only on type 2 receptor. So while they act on type 1 receptor, there is hypothalamic pituitary axis activation, arousal, CNS activation, angiogenesis, and appetite suppression. And the ones which act on type 2 receptors, it lead to anxiolysis, angiogenesis, appetite suppression, insulin or glucagon secretagogue, and vasodilation. Now, oxytocin and vasopressin. The vasopressor effect of the posterior pituitary extracts was first described in 1895, and the potent extracts were named vasopressin. In 1953, oxytocin became the first peptide hormone to have its structure elucidated and the first to be chemically synthesized, leading to the Nobel Prize in Chemistry being awarded to Vincent du Vignaux in 1955. The human oxytocin and AVP genes are situated tandemly in a head-to-tail fashion on chromosome 20p13, separated by a several kilobase intergenic sequence. Oxytocin and vasopressin mRNAs are among the most abundant messages in the hypothalamus being heavily concentrated in the magnocellular neurons of the paraventricular and the supraoptic nuclei of the hypothalamus, which send axonal projections to the neurohypophysis. Neurotensin. Neurotensin was isolated based on its hypotensive properties from bovine hypothalamus in 1973. The human neurotensin gene has been localized to chromosome 12, 12q21. Neurotensin producing cells are found in the midbrain, ventral striatum, extended amygdala, lateral septum, and arcuate nucleus. The actions of the neurotensin are mediated by three receptors, the NT1, 2, 3 receptor subtypes. The NT1 and NT2 receptors are 7 transmembrane G protein coupled receptors, while neurotensin 3 it is a type 1 amino acid receptor with a single transmembrane domain and is located intracellular. Now the effects of various neuropeptides in various psychiatric illnesses. First one is depression. Plenty of neuropeptides have been implicated in depressive disorders ranging from substance P, corticotropin releasing factor, thyrotropin releasing hormone, vasopressin, neuropeptide Y, and galanin. Large evidence, as suggested by Prang et al., reports that the thyrotropin releasing hormone is known to modulate neurotransmitters including dopamine, serotonin, and acetylcholine. Given that depression is associated with changes in hypothalamus pituitary thyroid activity, studies have reported its positive correlation with overt hypothyroidism. Okay, we can see this is the HPT axis. The thyrotropin releasing hormone neurons are located in the periventricular nucleus and contain thyroid hormone receptors and respond to increase in thyroid hormone secretion with a decrease in thyrotropin releasing hormone gene expression and synthesis. This is a negative feedback that is seen in hypothalamo-pituitary thyroid excess. During acute stress, glucocorticoids stimulate T4 production directly in the pituitary thyrotroph. Chronic stress inhibits the hypothalamic pituitary thyroid excess through different mechanisms. First one is high level of glucocorticoids. They inhibit the thyrotropin release, THR production by the thyrotropin releasing hormone neuron. Second one, corticotropin releasing hormone produces outside the PVN stimulates somatostatin production by neurons located in the periventricular area, which inhibit the thyroid stimulating hormone production. And third one, the glucocorticoids, these reduce the conversion of T4 to T3 by inhibiting D2. Studies demonstrated a significant reduction of elevated CSF corticotropin releasing factor concentration in patients with major depression who remained depression-free for at least 6 months following antidepressant treatment, as compared to no reduction in CSF corticotropin releasing factor concentrations in 9 patients who relapsed in these 6 months. This put forward the idea that poor response in major depression, despite early symptomatic improvement, may be showcased by elevated or increasing CSF corticotropin releasing factor concentrations in the course of antidepressant treatment. Hence, efforts are being made for the development of small molecule corticotropin releasing factor 1 receptor antagonists that can effectively penetrate the blood-brain barrier, as hypothesized by the fact that if corticotropin releasing factor hypersecretion is a factor in the pathophysiology of depression, then reducing or interfering with corticotropin releasing factor neurotransmission might be an effective strategy to alleviate depressive symptoms. Hence, the development of CRF1 receptor antagonist, now called as CRF receptor antagonist, that can penetrate the blood-brain barrier is a new class of agent for the treatment of anxiety and depression. For example, R121,919. Now in schizophrenia, cholecystokinin has found its role in humans in terms of gastric emptying, gallbladder contraction, pancreatic enzyme release, and suppression of appetite. The interaction of cholecystokinin with cholecystokinin receptor 2 has been reported to inhibit dopamine release, blocking dopamine-mediated behaviors in the anterior nucleus accumbens, whereas the binding with cholecystokinin receptor 1 in the posterior nucleus accumbens is associated with opposite effects. Cholecystokinin agonists that may be active in the treatment of schizophrenia should be non-selective or cholecystokinin receptor 1 selective. Overall, there is also evidence suggesting that cholecystokinin may be involved in both affective and stress-related disorders. Neurotensin is closely related to the transmission of neurotransmitters in the mesolimbic, mesocortical, and nucleus accumbens pathway, which are the major site of dysregulation in schizophrenia. Neurotensins are predominantly located on gamma aminobutyric acid neurons, which release GABA or dopaminergic nerve terminals and thereby inhibiting their release. Postmortem studies in the brain tissue of schizophrenic patients showed a decreased expression of neurotensin receptors and also a decrease in CSF concentration of neurotensin compared to their controls. And after 4 weeks of treatment with antipsychotics, the levels of neurotensin increased with the improvement of symptoms. Thus, neurotensin may act as an endogenous antipsychotic-like substance and awaits the development of a neurotensin receptor agonist that can penetrate the blood-brain barrier. Possibly most importantly, both neurotensin neurotransmission and antipsychotic drugs enhance sensory motor gating. Now, panic disorders. Cholecystokinin decrease the dopamine level. Infusion of cholecystokinin fragment has been reported to induce panic in healthy individuals, and patients with panic disorder exhibit increased sensitivity to the cholecystokinin fragment compared to normal controls. Pentagastrin, a synthetic cholecystokinin agonist, dose-dependently produced increased blood pressure, pulse, HPA activation, and physical symptoms of panic. Recently, a cholecystokinin receptor gene polymorphism has been associated with panic disorders. Autism. Oxytocin or arginine vasopressin neurons that are projections from the PVN to the forebrain and brainstem regulate learning, memory, complex social behavior, and female sexual behavior and facilitate maternal behavior. Cerebrospinal fluid studies of oxytocin levels in person with early exposure to stressful life situations showed a reduced concentration of oxytocin, indicating dysregulation of this peptide in psychiatric illnesses like autism. Recently, it has been demonstrated that recognition memory for faces but not non-social stimuli has been enhanced in humans by intranasal administration of oxytocin. Insulin-like growth factor 1 promotes a decrease in intraneuronal chloride ion concentration, leading to hyperpolarization in neuronal membrane and a subsequent decrease in neuronal hyperexcitability. This physiological effect has been considered to be behind the relative efficacy of bumetanide in improving symptoms of autism spectrum disorder. Dementia. Corticotropin releasing factor seems to be significantly reduced in cortical areas of patients with dementia. Reduced concentrations have been described in several areas of brain, including the frontal and temporal cerebral cortices, the amygdala, and the caudate nucleus in patients. A diminished concentration of temporal and occipital neocortical corticotropin releasing hormone was found also in dementia elderly patients. Corticotropin releasing hormone receptors are reciprocally upregulated in those cortical areas where corticotropin releasing hormone is reduced. The concentration of somatostatin has been investigated either in postmortem human brain material or in the CSF of patients with dementia. These reports convincingly show that there are reduced concentration of somatostatin in cortical areas of the brains of dementia patients. Personality disorder. A critical role of neuropeptides, including oxytocin, opioids, and vasopressin, may also be implicated in borderline personality disorder due to the involvement of these systems in the regulation of affective behaviors. Barbara Stanley and Larry J. Sevier propose a model of reduced basal opioid activity in critical limbic circuitry, including the amygdala and cingulate cortex, in individuals with borderline personality disorder. A behavior that occurs frequently in the context of borderline personality disorder and dissociation, non-suicidal self-injury, has also involved the implication of endogenous opioids. Now, addiction. Neuropeptide systems may modulate excessive alcohol intake based on evidence that ethanol drinking and energy balance influence numerous neuropeptide systems, and that manipulating these systems similarly influences ethanol intake. Neuropeptides in the hypothalamus can act locally or project to other regions to link with other systems, such as mesolimbic dopamine reward system, which regulate the reinforcing properties of natural rewards and drugs. More recently, the orexin system has found its role in drug seeking with several groups describing medications of alcohol, food, morphine, and cocaine intake via orexin A. Corticotropin releasing hormone being involved in moderating responses to stress is well established, and several studies have implicated this peptide in regulating alcohol seeking behavior. These are my reference. Its receptor is NK1, and its functions are it is one of the first responses to stressor. So, firstly, it may cause vasodilation with the nerve, and it is also present in amounts of the area posterior to the So, it is said that the post of nausea vomiting, NK1 receptor antagonist it would help in that. Apart from that, since it is increasing stress and depression also, so it has been seen that these NK1 receptor antagonists they have some inefficacy as in antidepressant.
Did you come across any medication specific to yes, presentation was good. Neuropeptides are there was a lot of enthusiasm about that. The name also used as that one neuropeptide has one function. It was initially thought and then we can devise medication to target that. We have come a long way. Earlier on, like there were tests, cases, so stress response used that if antidepressant, like maybe we can use that to diagnose the like hormones related to that. Now we know that these illnesses affect each other. So psychiatric illness have a physical influence also. We know that depression, schizophrenia, these patients have increased mortality secondary to like infections, AMI. So there are some mechanism by which they are affecting the immunity. Maybe neuropeptide is one. Second is physical disorders. They cause symptoms like low appetite, suppression, sleep impairment. So here again, we are like maybe if we have specific medications for that, we are maybe they will target us. The problem is that like the recent research that is coming is again that it is not like they are so specific like we were thinking that give one molecule and one target symptom, but they are acting in one more than one cases. So even for endocrine reason, if you give them it somewhere else. So that is something. We're still evaluating. Orexin is also like, I think orexin, some drugs targeting orexin system are used in anorexia also, because in that time it was withdrawn because of side effects. You said about like there are medication underway, but again, as we study them, we come to know that it is not as simple as targeting one. That's the thing.
That's something are not having drugs for appetite.
Percent. Yeah.
On the side people they were saying report that some cardiac side effects.
Suicidality, things like that. So, like you give medication targeting one symptom, then naproxen in that is
Effective in the patient feedback.
safety profile is good. So maybe all of this list of these huge list of neuropeptides, some may be effective. You may be able to specifically have medicines. For example, for insomnia, we have 10%. We may have for say pain reduction if it is there, as you said for substance P and all. It enhances the moment that it so the opioid with pain ka problem. That is major thing. Like you stop opioid because the endogenous opioids are they are suppressed, so there is greater pain perception. That bothers a person, some people very much. So if you can have specifically molecules that will increase that, so maybe we will have a better role in specific patient population of depression. Sorry, opioid substance. So dementia again. So there are number of areas where we can have research. So definitely some of the say preventions will come out specifically targeted.
You mentioned so many important things, autism, personality disorder, sleep and everything. Maybe in some years you might have
Oxytocin and autism. Oxytocin, autism.
Personality disorder, I think that I think say India में नहीं
It's oxytocin.
Oxytocin.
I'm not sure.
ये मतलब oxytocin कहता है just for research purpose or it is available there. Autism. It is like it has been demonstrated. It is like maybe used in research. Yes, ma'am. No, ma'am. This topic is interesting. Neuropeptides सबको can explain in detail. So we just to cover the basic things.
Oxytocin is like again much studied that social behavior it's not only for female, even like behavior changes after parenting, behavior changes of men also. So there also. It's not like you always oxytocin
Yes, and all.